Well, welcome in today. Good afternoon. This is the Colorado Fruit and Vegetable Growers Association Lunch and Learn webinar, Peeling the Onion, Growing Practices that Increase Salmonella. I'm Adrian Card, State Specialist in Produce with Colorado State University Extension, and I'm a founding board member of the Colorado Fruit and Vegetable Growers Association and your host for today's webinar. This is produced in collaboration with CFEGA and will address which dry bulb onion growing practices increase contamination of the crop with Salmonella and potentially other human pathogens. CFEGA is the go-to resource for Colorado's fresh fruit and vegetable farmers. We connect Colorado growers with industry, government, academia, and consumers to strengthen and expand Colorado fresh fruit and vegetable production. Learn more at the website and on CFEGA's social media channels. CFEGA is made possible by generous donations from sponsors, and the board would like to acknowledge its sponsors today. Here are the platinum sponsors, the gold sponsors, silver sponsors, more silver sponsors. Thank you all so much for your sponsorship of the association. Please learn more about these vendors at the website on the Grower tab, and you can peruse their products and services available to growers. We'll have Q&A at the end of today's webinar and look for a poll for educational outcomes as well. Our presenter today is Dr. Eduardo Gutierrez-Rodriguez. Eduardo is an assistant professor in food safety systems in the Department of Horticulture and Landscape Architecture at CSU. Dr. Gutierrez-Rodriguez has over 20 years of research, extension, and business experience in food safety, focusing on fresh produce and minimally processed food environment. His research centers on understanding the microbial ecology of human pathogens within farm and food processing environments, looking at microbial interactions, risk assessment, and remediation practices that can sustain fruit and vegetable production and reduce the transfer of human pathogens into the food supply chain. Eduardo, thank you so much for being here with us today. And with that, I'll pass over to you.
Thank you, Adrian. Good morning to everybody. Thanks for joining the webinar today. Let's see if I can show my screen so that you can all see my presentation. We're going to do this short and sweet so that we can hopefully open up for questions of the audience. We want to have a discussion here, and hopefully some of the information that we might present to you today will be helpful here. So like Adrian mentioned, we're going to talk about microbial risk profiles in onion production practices. I want to start by thanking those that have made this possible. I'm going to start by giving a big shout out to the master student Griffin Carpenter that sort of put quite a bit of work into these activities, and then the Specialty Crops Block Grant Initiative that has provided a significant amount of funding, and then of course Fresno State University, which was our collaborator, and Susan Pheasant and Jeffrey Dervision. And also want to make sure that I acknowledge folks from the Arkansas Valley, JB Ma and Kevin Tanabe for their support, as well as growers, Petroco Farms and Sakata Farms that have helped us quite a bit in some of these other folk activities. There are other partners too, but the Petroco and Sakata Farms have provided quite a bit of input. And then are the South folks, Sky Handyside and Hannah Sutherland, that have helped us quite a bit with some of the activities that happen at the farm. And of course, Tony Patton, Mark Uchansky, and Marilyn Drake that have also provided invaluable information and suggestions about the project. So want to make sure that AO acknowledge, and I don't want to forget the National Onion Association, because we've had multiple conversations with them too back in the past, and that sort of these projects spirited from some of these conversations and some of the listening sessions that we had with them. So I just want to make sure that you all know how much I appreciate you and how much it means to us, all the support that we have received for this time. So with that, we just want to sort of have a brief discussion of what we've been doing in the past 2 years. The picture you see on the right is one of the white onion cultivars that we were passing over at the packing line in Fresno, California. But why are we doing this? I think I always try to ask myself, why am I into this? What do we know about this? What are we trying to find? And hopefully what kind of suggestions we can have for you guys. So the spirit of the presentation here will be basically that. And so as I ask myself that question, why are we doing this? And we're doing this because before 2020, dry bulb onions were considered to be a very safe crop, right? There was pretty much not a single outbreak associated with dry bulb onions until then. Everything was associated with fresh cut onions or aspects that relate to processing in a fresh cut facility, which then lend itself for cross-contamination and therefore contamination of the onion itself. But before then, we never had an outbreak. But in 2020 and 2021, we had significantly outbreaks with Salmonella, which was the interesting character here. Two different types of Salmonella, Salmonella Newport and Salmonella Oriendenburg. The Salmonella Newport outbreak came from California, while the one that we were talking here for Salmonella Oriendenburg came from onions that were grown in Chihuahua, Mexico, okay? And so the key aspects that I think are important to highlight here is that in 2010, the National Onion Association had published some good guidelines about production practices in onions. Since then, there hadn't been an update of these guidelines. The two outbreaks happened in 2020 and 2021. The onion industry was already working a little bit on this. And so recently, in 2022, there has been a new update on these guidelines for dry bulb onion production. And they have done a significant effort to highlight some of these challenges in production. But we personally think that there's quite a bit of additional information to risk assessment and trying to understand the risks at the farm that can lead contamination of the onion and then to the consumer. And so with this research, what we've been trying to do is try to shed light a little bit into that aspect of things, okay? So what did we know about the risks of contamination before the outbreaks, okay? Most likely that we had three main methods of irrigation, which it's pivot irrigation, sprinkle irrigation, or furrow irrigation. And within that irrigation approach or system, we could have water canals or open water sources that would bring the water to these locations. And therefore, it was understood or perceived until the outbreak of 2020 that the highest likelihood of contamination could happen through the irrigation system. Some work had been done with E. coli to see if the irrigation system would be able to transfer some of that E. coli to the onions. And there was some evidence to do that. But overall, the understanding was that most likely the highest risk of contamination would have happened through the irrigation system. Then if we talked about what are other sources of contamination, one of the other practices or considerations was if I have some sort of disease, if I have some sort of plant pathogen disease in the field or at harvest or during storage, would that disease then allow for the nutrients of the onion to become available for a human pathogen, right? Many of these plant pathogens that we have macerate the tissue, allow for many of the nice sugars and acids and other components of the onion to ooze out of the onion in those moments of decay. And so understanding whether those situations would correlate with the presence of a human pathogen seemed to be important. From the 2020 outbreak associated with onions from California, one of the comments produced by the investigators, from the FDA investigators, was that there was a heat wave in the area and that the quality of those onions might have been slightly compromised. So could that be a situation just of stress, field stress, or could that be a situation of actually after harvest, those onions being significantly stressed and therefore providing the conditions for contamination? We don't know that. And so this is one of those unknowns that we had available until that 2020. The other aspects that we were talking about is what would happen in the field and what were those missing gaps of information that we would like to understand. So one of them was if you're doing some sort of topping, as you can see here on your figures on the bottom left, or if you're lifting on your bottom or your top left, or if you're putting into burlap sacks, are there any of those injuries or the cleanliness of those burlap sacks or the mechanized activities that pick up the onion from the field and put it in the big bins of tractors that bring the onion back to the packing house? Would those be sources of contamination? And at that point, and I think continuously right now, some of these is missing information specifically associated with burlap sack and management of those burlap sacks and cleanliness around them. And with this grant, what we have tried to do is look at topping and lifting and whether those could become sources of contamination. Another aspect that I think it's important is what kind of, does the scale color of the onion matter? If you're a purple onion, or if you're a white onion, or if it's a yellow or a sweet onion, does it matter? We know that there have been some anecdotal information that potentially onions might be able to inhibit growth of human pathogen. There is actually a paper from, I think it was Yugoslavia, that did some basic inhibition tests with some onion slurries, but it wasn't clear exactly what it is. But we know that the metabolites within onions have antimicrobial effects. And so does it matter if you're red? Does it matter if you're white? Would any of those inhibit growth of a Salmonella strain? So that's one of the things that it's also unknown, and we try to address here in the grant. And the other aspect that we wanted to sort of look into is the curing process. If I do a field curing versus a storage kind of curing scenario, is the survival going to change based on those activities? And again, the paper that was done in 2000, if I recall, 18 from Oregon State University that was looking at E. coli, at least seemed to suggest that at least in the field, it was not going to be a significant risk. So part of the understanding of why it was supposed to be a low risk crop was based on that study. And so in this one, what we tried to address was, if there is close contamination at harvest or during the curing stages, can that actually translate to storage? Can that actually translate to transport? Can that actually translate into the packing house? Because even though we have the 2022 guidelines available, that information continues to be absent, okay? And then finally, I guess by far one of the most critical ones that was highlighted in both investigations outbreaks from 2020 and 2021 is the cleanliness of the packing house. Do we have clean breaks in the packing house? Are we having wet sanitation? Are we having dry sanitation? Do we have any kind of sanitation at all? And trying to understand whether those dry scenarios where there is a lot of debris coming from the scale of the onion or from the residues of the onion, are those going to increase the survival and persistence of a human pathogen? Can those actually serve as cross-contamination as the onions are rolling, as you see here, where these ladies are selecting the onions, or down here in the conveyor belts that are sort of separating the onions based on weight and color, whether those specific areas that are rarely clean become a source of contamination. Because today we want to cap it only in field production practices. We're just going to talk about this today. In the next webinar that we're going to have, I think 1 week from today, then we're going to focus all our efforts in the packing house because we have tried to look into this project too. So again, just to be concise and to make sure that we're all in the same page, what we're going to talk about here today is only specifically to field contamination events. And so to sort of succinctly try to put this, what we need to, or what we are trying to find out is whether harvest and storage practices, or even the variety or the season, impact the contamination of the onion during the cropping cycle, at harvest, during transport, and during sorting and packing in a packing line. That's basically the overall need of the industry. Back when we started this project, we continue to believe that that need is still here. And that's what we try to address with this grant. So now let's sort of dive deep a little bit more into what we found and what we think it's the overall message here. So does it matter to be red, white, or yellow? Is the onion going to have the necessary inhibitory capacity to inactivate a human pathogen? So we grabbed 17 of the most important onion cultivars grown in Colorado and in other regions, and we evaluated whether those different cultivars will inhibit Salmonella. So we had red, white, and yellow. And what I'm going to show you here is the result of 10 of them. Okay. So if I were to just succinctly tell you whether the onion inhibited Salmonella, the answer to you will be no. Okay. So I'm going to show you now a graph that is a little bit busy, but the message that I want you to come up with is that it doesn't matter if I'm a sweet onion, if I'm a white onion, if I'm a yellow onion, if I'm a purple onion. What it looks like is that irrespective of that, the constituents of the onion, even though they are diametrically different, they don't inhibit Salmonella. Okay. So what does it mean for a grower is that I need to focus on the practices in the field, because it doesn't matter what kind of onion I do, I'm going to have the same risk on any kind of variety. Okay. So if we go into much more detail here, you will see that on the y-axis, you have the population of Salmonella, how many Salmonellas we have. And on the x-axis, we have different cultivars of onions. As you can see, some of them are white, some of them are yellow, some of them are purple. So these blue bars mean the population of Salmonella before we started the studies, okay? When we put it in the onion, in an onion slurry. So we had 1 million cells. That's basically what we had. And then what we wanted to see is that if the Salmonella was swimming in that onion slurry, which will have the direct nutrients available to the Salmonella, if those will die or grow or stay the same after 24 hours, because that is the most direct way that the Salmonella is going to be exposed to. When the onion is harvested, you have the scales, you have all these other things that will prevent it from getting access to it. So if the Salmonella responds to that slurry, then we're going to know if it happens or if nothing happens. So when we did it after 24 hours, which is these orange bars here, what you can see across all the cultivars, is that the population of Salmonella pretty much stayed the same, and in some cases slightly grew. So that's what we're saying, that even though we had this significant amount of Salmonella and this significant amount of onion constituents available to the Salmonella basically didn't die. It just stayed the same or slightly grew. So when we got this information, then we said, okay, so when we're going to put our field trials, what we're going to do is that we're going to grab a purple, a yellow, and a white, because those three were implicated in the outbreaks. And we're going to taste whether the field activities that I do increase the contamination, knowing that right now, based on this information, it didn't matter what kind of cultivar you were. Okay. So this is an example of the field studies that we did. So you can see Griffin, which was the student here on the left, with his Tyvek suit and sort of walking around the field. If you see these little white sockets down here, that's where we put the Salmonella. It's kind of like a tea bag that has the Salmonella inside with some sand and some milk powder that allows it basically to even contaminate the field, okay? And then we did thorough irrigation, because it will be the method that allows us to basically evenly contaminate the field. Why is it important for us to evenly contaminate the field? Because then we can decide whether we don't recover or recover Salmonella, not because we didn't contaminate it correctly, but because there are environmental factors that are impacting the survival of that Salmonella. So if you look at these towers here on the top right of your figure, we had a weather station in which we basically were recording all the parameters. Also, Griffin did a great job there. And what we had is different moisture probes, UV light sensors, and all these things, because we wanted to know whether moisture content or UV light also may have an impact, okay? So that was basically the experimental setup. To be a little bit much more detailed about it, we had 3 field replicates across the fields. Each replicate was 100 feet long, so we had a significant amount of onions. And if you see here on your right side, these circles, basically it indicates the locations that we were sampling within the location that we put the Salmonella. So we had a very robust amount of samples collected over time. Okay, just to sort of finish it, I love this picture because we really did a great job here in the distribution of the fields. And so that is how the red and the white onion cultivars kind of looked during the growing season, and how we were segmenting the inoculation so that there was no cross-contamination. So let's think about it this way. The simplest question that I get a lot is, how long does the Salmonella survive in soil? How long can it be there? Okay. So we started at a population of about 100,000 cells, which is this log 5 that is on the top left, okay, 100,000 cells. And we basically went and collected soil from where? From red onions, from white onions, and yellow onions for 64 days, okay? So if you see in day 1, we had about 100,000. In day 4, we had about 1,000. On day 8, we were below 1,000, and it just kept going down. And by day 32, we were about 100 cells every time that we collected the soil. You see that between day 32 and day 34, there is a gap, and there is a reason why behind that. But what I want you to all understand is that at the end, after 64 days, when we were ready to harvest, the population of Salmonella in those crops was about 10 to 15 cells per gram of soil. Okay, so it was able to survive over that period of time. And at the moment of harvest, we had a very small population, but it still was available and we were able to recover it. Okay, so if we were to just talk about, does Salmonella survive along the onion cycle in soil, then the answer will be yes. If there is contamination, it would probably stay there for the entire cropping cycle. And depending on how much population you have, it may die off faster or slower. The other thing that we wanted to see is what happens during topping, which is a typical practice of onion production system. So we went ahead and topped the onions. We inoculated again the onions and looked at how did the contamination happen. So topping happened at day 47 within the entire cropping cycle. And we did another inoculation there so that it was at the same inoculation at the 3-leaf stage. And what you see here in the next 16 days is that there was a slower decline in the population. Why? Because the UV index during that time, it's much lower than at the beginning of the 2-3-leaf stage. The UV index at the 2 to 3-leaf stage was about 8. Basically, you will become crispy. But if you are in the topping area at that sort of late season, then the UV index is 4 or 3. So it has basically decreased a complete amount or reduced by itself by half. And the weather is much more mild. So then what happens is that in here on the 2 to 3-leaf stage, you see a crisp decline, but on the topping aspect of things, it just goes much smoother. And so by the time we get to harvest, we have almost 1,000 cells of Salmonella in 150 grams of soil. So basically we have 1,000 cells at harvest and topping versus just 10 cells at harvest when it happened at the 2-3-leaf stage. So what is the message here? The closer does the contamination happen to harvest, the likelihood is that the contamination is going to pass to the onion at a much bigger population. We tried to do this too also with top lifting, because that is a significant practice too. And again, it happened within that period of 47 days all the way down to day 70. And again, what you see is also a very small decline, and again, a very high population of Salmonella at harvest. If you just look at this graph, you will see that the purple, yellow, and gray bars, they're pretty much the same. So what is this data telling us? That what we saw at the beginning, that the onion cultivar doesn't matter, also applies here in the field. It doesn't matter if you're a red onion or a white onion or a yellow onion. If the contamination happens early in the season or late in the season, the Salmonella is going to stay in the soil along the whole cycle. And the closest that you get to harvest, then the populations will stay much higher. So if I am a grower, I need to think of the risk. And so I need to make sure that as close as I get to harvest, that there is not going to be any kind of contamination event, because if that happens, the risk is going to be high. If the contamination happens at the beginning, at the 2 to 3-leaf stage, the risk is going to be lower. Why? Because by the time I harvest, the population is going to be small, but it doesn't mean that it's going to go away. Okay? All this information that I've been sharing with you is associated with soil, which means soil, Salmonella is going to stay there for the entire season. Now let's talk about the population of Salmonella in the actual onion. Okay. So here is the population of the Salmonella in the actual onions. This is at day 1, day 4, day 8, day 16, all the way to day 64. Again, you see that all the bars are pretty much the same. There's really not a big difference between red, white, and yellow. But the population stayed consistent, at least for 32 days. So we had log 2 means that you had about 100 cells at the beginning in the 2-leaf stage. And by the time that you got to harvest, when you had the full bulb ready to bring over to the storage area, we still had about 10 cells, or between 10 and 100 cells, which is what you have here at 64 days. So where is the contamination higher? In the soil. Where is the contamination lower? In the onion. But if it's still in the onion, it will stay in the onion until harvest. So the concern is, if my soil is contaminated, could I cross-contaminate the onions? And the answer is most likely yes. So the goal here is to see how can we prevent cross-contamination of the soil that would lead to contamination of the onion. Irrigation system, the way that we used it, it will be a simple way of doing it. So if we were using if we were using water with doubtful microbial quality, meaning with water that is compromised in their microbial quality, it will be pretty easy to contaminate the entire field. Okay. So if we were to sort of put it together, summarize it all together, this is how it would look like. So on the top 2 figures that you see is basically the survival of Salmonella from the 3-leaf stage all down to harvest. On the left side is is soil. So you see a sharp decline in soil populations that then stabilizes it. And in the case of onions, on the top right, then the population basically is consistent across the 64 days of production. If we move down to the topping and lifting stage, again, what we say is, oh, I had a high contamination event in those stages. I am close to harvest, 15 to 20 days. That contamination is going to translate to the onion, and the populations are going to be high. What we did see was a slight difference in the population between lifting and topping. We don't want to make a big deal about it because statistically speaking, there is a difference, but biologically speaking, we question that. And what do I mean by that? Here on the right bottom side of lifting, you might have like almost 4 logs or 10,000 cells, versus in the topping side of things that you might have 1,000 cells. We do think it's more an artifact of sampling, not an issue specifically to topping or lifting. But what we want to make sure is that you understand that if you harvest that onions and we had that late contamination event, those onions are going to have the Salmonella at high populations and are going to transfer it to all the equipment and other aspects that you have downstream. Okay, so the last 2 slides that I have for you before we open it up there for questions is, once we harvested the onions, and again, I want to remind you, I'm going to go back here where it says, when we harvest the onions from the 3-leaf stage, we had about 10 cells, which is this top right figure. When we harvest the onions from topping and lifting, we had about 1,000 cells. So we will have a low risk situation when the contamination happened early in the season, and a medium to high risk when the contamination happened at late in the season. So once we harvest that, then the next step was we need to put it in bins, we need to store it, and we need to send it to California so that we could see what happened with those populations. Okay. So this slide basically shows you how we put the onions in these card boxes. The tractor basically lifting the card boxes and putting them in this semi that was bringing the onions back to California. And then the final figure here on the top bottom part is where we have all the onions when they arrived in California. So if you remember from what I told you before, at the beginning of harvest, we had between 10 and 1,000 cells. But when we arrived to California and we were going to start the process, as you can see here, we had less than 3 cells. So between the moment that we harvest them, we put them in those bins and we ship them over to California, which was about 15 days altogether. The Salmonella continued dying and it went from 10 to 1,000 cells to less than 3 cells. So during that time, the conditions were not favorable for that Salmonella to continue growing or staying there. We were not expecting for such a significant die-off, but that's what happened, okay? Next week's seminar, we're gonna talk about what happened in the packing house and all the different interactions we did. But to sort of succinctly finish here the presentation, these are the 4 things that we wanna share with you today. Any kind of cultivar or any kind of scale color don't seem to impact the survival of Salmonella, meaning you need to focus all your efforts at the farm, at preventing contamination at the farm. If the contamination happens in the soil, most likely it's going to stay there. And unfortunately, we don't have any remediation practices right now to eliminate the Salmonella from the field, okay? So if you were to have it there, most likely it's going to basically stay there and cross-contaminate the crop. When we are doing late season activities like topping or lifting, there is a higher chance of contaminating those onions. So that means that if my soil was contaminated and I start doing those activities, I will be able to transfer that contamination to a higher rate to those onions. So again, going back to the original message, we need to try to prevent that contamination when we're preparing the field, when we're planting the onions, when we're starting to do the irrigation, because if we prevent that contamination there, then the risk is going to be low. But if we don't, then the risk is going to increase exponentially as we get closer to harvest. The good thing here is that even if we had high populations of Salmonella, 10 to 1,000 cells, like as we see here in this last bullet point here, during harvest, storage, and shipping to California, during those 15 days, we did see a significant die-off of that Salmonella, which could help. But that doesn't mean that the Salmonella is not there. And that doesn't mean that that Salmonella can then transfer to your packing house, okay? So even though that can happen and that decreases the survival, we can still have a significant risk. So the goal is to try to have as little as we can in those onions so that if anything is there, we will be able to use sort of that advantage of timing and processing to further stress the cells and reduce the population. But we are at the mercy of the fitness or how strong that Salmonella strain could be. And so with that, I want to sort of stop here and thank everybody for your attention. We have pretty much 10 minutes for questions and comments. And again, thank you so much.
Eduardo, fantastic presentation. Tons of great information in there. Thank you. Looking still for questions in Q&A. And before we see some of those, I just have a very high level question on this. If we think that in your research found, you presented that in storage and transportation, there is significant die-off of Salmonella. How do we explain, what do you hypothesize are the conditions that created the outbreaks in 2020 and 2021? Is it a short window between harvest and marketing where it hits the produce floor? Any thoughts there?
Sure. That's a rough one because of several things. Historically, there hasn't been dry sanitation activities or sanitation activities in most onion packing houses. There's been different levels depending on where you are in the region. Some are much more paying attention to those situations, some are less. So and the onion industry has been considered safe for so many years.
Mm-hmm.
So from the investigation that the FDA did, there was clear evidence that something in the packing house could have led to that contamination. So if you're building accumulation of debris or residues, onions, or microorganisms at some point in the packing house, you're going to have a high population that could become a risk of contamination. This is pure speculation from me based on the FDA investigation, okay? If we think about activities that can happen in the field, in the outbreaks, they had 2 commonalities. Areas are with animal operations around them. Areas had weather patterns or issues with it. So those could have been conditions that would lead to potential cross-contamination of those onions. And if there are issues of sanitation in the packing line, then the risk becomes much higher. From what we did, I was pleasantly surprised that we had such a big die-off of the Salmonella during harvest, transport, and storage. I was not expecting that. But remember, the Salmonella, if it's going to be in the onion, is going to be on the outside. So there is not enough nutrients outside. And then if you are dry, you're covered, you don't have the necessary environmental conditions, then these organisms could die faster. Okay, so that's kind of like my spiel about around that. So the question is, if it's low, can it be transferred to the packing house? And if it's in the packing house, can I do anything in the packing house? And so hopefully we're going to talk about it next week a little bit more in detail.
Fantastic. Fantastic. We got a great number of 2 questions coming in. Great questions here. Well, first from Joe. If the water in organic production is using irrigated subsurface drip lines, water is untreated, but it is filtered, will the onions take up Salmonella through their roots?
Awesome question. Thank you, Joe, for the question. There is not a single piece of evidence right now, research-wise, that shows internalization of human pathogens in onions. There is significant research in green onions, but the difference with green onions is that, of course, it's a much shorter cropping cycle, and the green onion is, it's still green, it's still small, and there is much more likelihood of contamination because it's being harvested by hand and you're making these bunches and you have all these other aspects. But even in that scenario, they haven't been able to show that the human pathogen internalizes the green onion. What it has been demonstrated with the green onions is that cross-contamination and the cutting that happens in the operations at the top and then at the root system is the one that leads to cross-contamination of the onion. That, if we translate that to dry pulp onions, is similar to what? When we do the topping, when we do the lifting of the onion, those are the moments in which onions that were intact could get contaminated if the soil or the surrounding areas has that contamination. Now, you're using drip. So you filter the water, but if you're not treating the water, the filtering is not going to do anything to an organism. It's just going to be there. The drip is going to be basically next to the bulb. So if we were to hypothesize that the contamination happens at that dripping area, then that would be the high risk area when you lift or when you top the onion at that cross-contamination situation. I know that irrigation and drip irrigation is a little bit of a cumbersome thing depending on the water source that you have and how you can go about it. And, I've been asked many times about treatments, and that's kind of like a whole different seminar about it. But if I were to say some additional comments around this is that by minimizing the amount of water that you have around the bulb, the less likelihood there will be to spread that contamination evenly in the field, like we did with furrow.
Great. JB has some questions here really related to monitoring and best practices. There are 3 of them, and I'm going to start with the first, since we were just talking about soil. He asks, how often should growers monitor food pathogens in the soil, every week or just before harvesting? Do growers even do that? That's not a thing yet, right?
Okay, so I'm gonna go with number 1. What is the rejection threshold for food pathogens for onions? Basically, 0 tolerance. There is no tolerance. If you have positive, shouldn't go to commerce, period. Of course, if the positive is in soil versus if the positive is in the edible portion of the crop, there is some leeway there, right? But the idea is that if it's in the crop, done. If it's in the field, then there are other things that you could do about it. Most people that I know, not only in onions, but in fields in general, don't tend to do a weekly sampling of soil or things like that. In the leafy green industry, there are companies that before they plant the crop, they do a field assessment, including soil sampling for the presence of human pathogens. What I know from them is that if they have a positive in soil, they don't grow the crop there. But there is not established treatment frequency or anything like that. If we think about good agricultural practices, which is what most of the things we're doing here, the focus, like I said at the beginning, is looking at the history of that field. What happened in that field before I put my onion crop? And what am I doing in that period of putting my onion crop that could increase contamination? That is where the grower, I think, in my personal view, has to put the biggest money or attention. What was happening in that field? And what am I doing at that moment so that it can prevent cross-contamination?
Mm-hmm.
And so then number 3 is, if there are foodborne pathogens in the field, that is the million-dollar question. What can a grower do? All right, so right now we don't have bioremediation or remediation practices in soil. We don't have them. I know people that have tried methyl bromide, which is already banned, and it didn't work. There are practices that we have used, and some of them are published already, that include solarization and the use of cover cropping. But if just you think about it, if you have 100 acres and you have to solarize 100 acres, it's not feasible. It is just not feasible, okay? So I think the next frontier, or the next aspect that needs to happen here, is what can be feasibly be done in a field to remediate it if we have a human pathogen. Like I told you before, JB, those growers that know that there is a field that it is contaminated, they leave the field and they don't use that field. What do? Anecdotically, what they've done is that they disc the field, they irrigate the field, and they try to do these dry wet events for 3 months, and hoping that there is that dryness, wetness, desiccation situation is going to expedite die-off. I have experiences with crops that have been, or fields that have been contaminated since 2012 with Salmonella, and they're still contaminated in 2022. So that is the problem that we have. We don't have a practice yet that we can use.
And there, I'm seeing emergence and conversations through Western growers of really beginning to look more broadly in pest control, but in this case, microorganisms and food, produce, towards biological solutions. That's the mindset a lot of organic producers come to this with. Are we seeing any solutions there that would be soil treatments that you're aware of? They're in trial, or?
I mean, sorry, I have to put a plug here. I'm working with a company that is developing some novel sanitizers that are GRAS, meaning generally regarded as safe, that have shown some significant die-off in soil, that you would basically kind of do like a pre-plant application, kind of like if you were doing a lime application, kind of like in the same scenario, that basically would work with the pH and would work with the inactivation of the organism. We've seen some important reductions, but of course we have so many different soil types that if we don't know if you have a very high loamy soil or high clay soil, if it's going to respond the same.
Right.
We've seen people do cover crops. Cover crops is something that has happened for centuries, and so the allelopathic effect of these has been shown to work with some plant pathogens. We have done some work with human pathogens, and there are some mustard crops that seem to have it, but again, it's only in vitro or in very controlled scenarios. When we go out in the field, we don't seem to have them. And to finally address your question about microbial diversity in organic systems, there are mixed publications. Some of them say that microbial diversity does inhibit some of those survival of human pathogens. There are some others that actually say, nope, it actually supports the growth. So I think that one of those other frontiers is looking at organic systems and what practices may or may not support the growth of human pathogens.
Great. Well, Eduardo, we'll leave it there for today. Mark says thank you. I say thank you for keeping us on the leading edge of these conversations and leading information, and lots of collaborators and colleagues in today's webinar. And I'm sure we all thank you for your leadership in produce safety at CSU.
Thank you so much. I appreciate everybody for coming. Take care.
Thank you, guys. Have a great day.